Tonight's presentation is a continuation of a trend that we started last week, a great trend that I'd like to finish the semester off. We hear from KSC students about their research and their work. Today we're going to hear from two students, Slesha and Carter, who are majors in the program Sustainable Product Design and Innovation. To introduce these students, I'm happy to give the floor to one of my newest colleagues, Professor Muhammad Hassan Habib. He's an assistant professor in the SPEEDY program and has been since 2019, long enough for him to become an important part of the campus community and long enough for him to begin to mentor students of all sorts, including the two that you're going to see tonight. When the students are done with their presentations, we'll open the webinar for questions. I'll return to help with that and rejoin Professor Habib and the two students to help close this out. So with that, I will hand it over to Professor Habib.
Hi, my name is MD Hassan Habib, and here I'm going to introduce my two research students. So one of them is Slesha Tuladhar. She's basically doing her double major in architecture and SPEEDY. She has been researching in our additive biomanufacturing lab since last year. And she has since she has math, chemistry, and CAD background, this really help her understand the basics of biofabrication. And she's mainly focusing on a very important topic related to biofabrication, which is rheological properties of biomaterial. And finding out rheological properties is very important to define the 3D printability of biomaterials. At the same time, which will consequently help take decision if we can provide that biomaterials as a safe house for the living cell. Recently, she has published two international conference paper in Institute of Industrial and Systems Engineers Conference and Manufacturing Science and Research Conference organized by American Society of Mechanical Engineers. And she has recently received a National Science Foundation Student Support Award announced by American Society of Mechanical Engineers. So Cartwright, or I normally call him Carter. Carter is another SPEEDY student, and he has been researching in our lab since last summer through NH Biomed undergraduate research training program. And his strong math, chemistry, and CAD background really helps him understand again the basics of biofabrication. And at that time, when he started, he was asked or tasked to design and prototype a nozzle system for multiple material cell type extrusion to help mimic the native tissue. Today, he's going to present a portion of that work. He also has published two international conference paper this year in the Institute of IISc, Industrial and System Engineers Conference and Manufacturing Science and Research Conference organized again by ASME. He also participated in various poster presentations like in INBRI Annual Meeting 2020, COPLAC 2020, and AEC 2021. And he has recently received the same award Slesha received, National Science Foundation Student Support Award. So here I'm inviting Slesha and Carter to present their research.
Thank you so much for the great introduction, Dr. Habib. I would go ahead and share my screen. Can everybody hear me? So again, like Dr. Habib said, I have been working for Dr. Habib in his additive biomanufacturing lab with Carter for about a year now. And today I will be presenting you about the research that I have been focusing on ever since. It's called the rheological analysis of hydrogel for 3D bioprinting processes. So this presentation is going to cover some of the introduction to what is 3D bioprinting and why it is important, what is rheology and what is shear thinning behavior and why it is important for 3D bioprinting, some of the things about biomaterials that we've been using and the procedure of how we got into making the biomaterials, the rheological analysis, in the form of flow curve, thixotropic test, amplitude test. We would also go a little bit into 3D bioprinting process and how the fabrication is done. Also, some of the future works that we are looking forward to do. And on your right, you can see a picture of how the rheometer looks like. It's the machine that I work with. So what actually is 3D bioprinting then? It is a very revolutionary technology that's been gaining popularity nowadays and uses a computer-controlled 3D printing discipline to release biomaterials layer upon layer along with high precision positioning of cells. So the way it works is given in the picture. You take cells or group of cells, you mix it with the hydrogel, and this is what we call the bio-ink. After setting a suitable parameter, you kind of like load it into 3D printer, and then with all this setting and parameters done, you extrude it out. Then it is incubated in proper culture for days, and that is how the tissues are regenerated. So why is 3D bioprinting important? About 122,071 patients are still waiting for organs, with 48% of them waiting for more than 2 years. So I think that if 3D printing, 3D bioprinting is ever possible in future, which I think is going to be possible in future, but if it ever is possible in the future, then I think it's going to be a very big breakthrough in regenerative medicine. The way the machine itself works is on 3 axes. It has 3 axes as given in the figure. It has a pressure controller so that it can extrude the biomaterials that we were talking about in and out. It also has a build plate where the scaffolds are extruded, or this is where we hold the scaffolds. Here is a small video of how it works. So this is the 3D printer working, and on your right you can see a model of the leaf that is being printed by the 3D printer. So going a little bit into rheology and why it is important for 3D bioprinting. Well, rheology is a field of science that deals with the flow of materials. Since we are working with biomaterial and it has certain kind of viscosity, it is very difficult for us to know how the material is going to behave while it's printing, before it's printing, and even after printing. So it is very important for us to know where the biomaterial is it is like what stage it is at in terms of viscosity, and this is what the rheology teaches us. There are 2 types of fluids in this universe, one being the Newtonian fluid. So the kind of fluid that do not change the viscosity upon the application of force are called the non-Newtonian fluids, for example, water. And there are the type of materials that change the viscosity whenever there is application of force. Whenever you try to put force on it or even pressure on it, they change the viscosity of it. And it is further divided into 2 types, being the shear thickening fluid and shear thinning fluid. So shear thickening fluid are the kind of fluid that increases its viscosity whenever you put more force on it. One of the real life example would be cornstarch. So whenever you put a lot of force into it, it basically acts as a concrete or a floor. And the other one is shear thinning fluid, which means these kinds of materials lower their viscosity whenever you apply certain kind of force or pressure into it. And the real life example of this would be ketchup. Whenever you are trying to like put force on the bottle to like squirt the ketchup out, it reduces its viscosity and it comes out of the bottle. Most of the biomaterials that we are using show shear thinning behavior, and it is very important for these materials to have shear thinning behavior. It is because whenever it's going through a very small nozzle, you know that these biomaterials are already treated with the cells, or they are already mixed with the cells. So whenever they are going through very small nozzle, it experiences a tremendous force. So if it has a shear thinning behavior or shear thinning property, it would be a little liquidous and helps the cell to breathe and helps further in the cell viability. So there's a better chance that the cells are going to survive. But then if we were to think about it, and if it extrudes out being liquid, it's going to be so hard for us to retain the kind of shape that we wanted to retain. So for example, just imagine we want to like print a nose, and if it is very liquidous, it's not going to retain the kind of structural integrity that we wanted to retain. So getting to know how these biomaterials work, how the viscosity is going to change on the application of force and a different kind of parameters is very important. That is what rheology helps us to understand. We can see in the picture like how low viscous natural polymers print out to be. They do not perform that well when you print it out. They tend to like disperse more, and it's clearly shown in the picture. Um, Meanwhile, on the contrary, that we can see the high viscous natural polymer, and we can see how it's like holding its structure after printed. So this is why rheology is so important. For this experiment in particular, we have been using two kind of materials, one being sodium alginate, which is a seaweed extract and a very huge molecule, like you can see in the chemical formula. And the other one is the carboxymethyl cellulose, also known as CMC. So the way we prepared this biomaterial is we kind of worked around having a percentage of solid content of alginate and CMC, so that we could see the effects of concentration on the viscosity of the material. Then we mixed it up together. We stirred it with magnetic stirrer until it was homogeneous mixture. Going further, we did 4 rheological tests under rheometer, namely the flow curve, thixotropic test, amplitude test, and frequency test. Then after doing that, the software produces a lot of graphs and curves that were very helpful to further predict how they are going to behave. And then the further analysis was done in the actual sheet, which is more like compare and contrast of, or even like getting average of how the multiple numbers of experiments turned out to be. So this is the first rheological test that we did. It is called flow curve. And this is the result of the flow curve that we did for the concentrations the compositions that we made. For this experiment, we made sure that the shear rate was 0.1 to 100. The shear strain for the amplitude sweep test was 0.1 to 100%. And for the thixotropic test, we made sure that we took 3 intervals of time, which helped us know how the biomaterial is going to heal after it shows the shear thinning behavior. So on the graphs, we can clearly see that the materials are showing shear thinning behavior. It is exactly what we wanted, and that is what the graphs are showing. On this slide, this is drawn from the amplitude test, and it gives us more knowledge about the storage modulus and loss modulus of the biomaterial. So the storage modulus of the biomaterial gives us an idea of more like a solid characteristic of the biomaterial, while as the loss modulus shows more of the liquidus characteristic of the biomaterial. And amplitude test gives us a lot of data involving storage and the loss modulus, and it also gives us the dilution point, which is like this sweet point. If it goes above that, it's going to be a solid, and if it goes down below that, it's going to be a liquid. Here is the thixotropic test, also known as the 3I2T test. On the graph on your left, you can see that how this biomaterial is going to act when it is at rest, when it is as retention, and when it is extruding the biomaterial, when it's like experiencing certain kind of force. And on the right of the same graph, you can see how the biomaterial is going to heal or kind of recover from its shear thinning behavior until it comes to a normal position or normal environment. For example, we can see that A0C8 on the graph down on the graph below, A0C8 showed 91.2% of recovery after 60 seconds and 96.1% of recovery after 120 seconds. After getting all these rheological data, we went further with doing or printing these compositions with the 3D bioprinter. We made sure that the parameters like raster width, layer height, nozzle speed, print distance, like even the path tool coordinates were set the way we wanted it to set. And on this picture, you can see that we are mostly working with the single nozzle system right now, but my coworker Cartwright Nelson, he is researching more on how to make this be the double nozzle system so that we can mix different biomaterials at a real time. Right now we are just mixing it beforehand before printing it, but his research focuses more on how can we do at a real time when we are printing it, how can we mix it at the same time. So these are the results of the printed scaffolds, and we can clearly see that A6C2 is showing a lot of diffusion than other scaffolds. Also, the filaments fabricated with A4C4 and A6C2 showed 48% and 77% more diffusion than filament fabricated with A2C6, respectively. Some of the future work that we are looking forward to do is print more multiple layered scaffolds, do a lot of cell encapsulation work, and also doing some of the cross-linking at the beginning, because most of the cross-linking that we are doing right now is at the end. So we are looking forward to doing these things, and thank you all for being here and listening to this presentation. It was a great experience. Before leaving, I would like to acknowledge Keene State College, the Sustainable Product Design and Innovation Lab, for giving us the space and equipments to work with, New Hampshire Biomade and New Hampshire INBRI for funding this project and also giving us a lot of equipments to work with. I would love to hear your questions and concerns about anything that has to do with 3D bioprinting, but right after Carter does his presentation. So I would like to call Cartwright Nelson to present his presentation. Thank you.
Hello, can everybody hear me? All right. Thank you, Slesha and Dr. Habib, for the introductions. All right, so my name is Cartwright Nelson. I'm a junior at Keene State College, and I am a sustainable product design major. And I've been researching a nozzle system that's able to interchange between materials without stopping its deposition for the 3D bioprinting processes. So Slesha already gave a little bit of an introduction, but 3D bioprinting is used to print living cells, and the goal is to hopefully print functional tissue or organs eventually. And the way this is done is through reverse engineering. So what would have to be done is the model, such as a liver or pancreas, would be scanned using a 3D scanner, and then a cloud point matrix would be generated, which would then be converted into a 3D model, sliced, and a slicing software would generate a print toolpath so that each layer can be printed. And one of the interesting parts is that the coding for this toolpath, which is usually G code, can be altered so that each scaffold has a different porosity. And we can fabricate things such as bones, which have different densities at different sections of the cross-sectional area. And we could also use this to customize functional tissue for individual patients. So motivation for our work. Right now, development of functional tissue is not possible, but the way that people are doing it is using multiple print heads because it requires a lot of different cell types and a lot of different biomaterials. And the challenge with switching between print heads is that it decreases the efficiency of the print, the structural integrity, and the accuracy. So this obviously isn't good if you're trying to print a tissue that you're going to implant into the body, because you don't want certain cells in places that they shouldn't be, or certain materials in places that they shouldn't be. And you also wouldn't want breaks in the deposition and just gaps in the print, because this has to be very accurate so that it can hopefully someday be used to transplant functional tissue and organs. So our proposition, I've been developing a nozzle system that's able to print multiple different materials with continuous deposition, and the goal is to minimize the transition distance to almost zero, if not zero, so that precise deposition of transition can be achieved. This has been tested in different ways, but the way I've been testing it is a spiral that I designed in Rhino software. Before printing this, the parameters of the print have to be set, such as raster width, layer height, nozzle speed, print distance, and pressure used to print. And one of the things to keep in mind is that the higher the pressure, the more damage is caused to the cells. So I've been doing my best to keep the pressure as low as possible, and you'll see how that affects my design process. The transition distance is the difference between the initial transition or material change and the full transition. All right, so my designs, I started over the summer just throwing stuff together by hollowing out a 3 milliliter syringe, running tubing through it, and sealing the whole thing with epoxy. I then modeled this in Rhino CAD software and printed several different prototypes. I think there were maybe 8 prototypes in total, because each one had its own issues. The first, which is the tube in the top middle, our in-house 3D printer, not bioprinter, at the time was having issues with resolution and printing the inner diameter of the nozzle small enough and also equal to the other diameter. So I ended up using an insert, which is seen below that, to increase this diameter. This worked pretty well, but the setup on the printer that you can see required preloading of material. So it was a big waste of material. And once the plungers in the syringes were at the bottom, it was unable to print, because even though there was still air pressure and material, the plungers couldn't go anywhere. So the whole thing was left full of material and had to be reloaded over and over again, which was very wasteful. It also required a lot of pressure, and the pressure would actually backflow material into the other side of the system instead of coming out of the nozzle. So in order to fix this, the design I'm testing right now uses medical components, which shortens that transition distance a lot. It also uses duckbill check valves, which keep the material from backflowing and allow it to deposit material much more precisely. This is also nice because it has a lure lock at the bottom, which allows me to use any nozzle diameter or type that I would like. And to test the transition distance, like I said, I developed or modeled a spiral and use this to, by measuring the print speed and the time it takes to transition, I can find the transition distance. So you can kind of see how it would be preloaded with one material, and then as it starts printing, it switches to the other material, and the time that this takes to clear all the way out and start printing the other material is recorded, and that is the transition distance. So to minimize this, I'm working on shortening that mixing chamber length, and also testing many different parameters. The prototypes I showed before, the first one required 120 kilopascals of pressure, the second 100, and the third 80. And then the proposed one here only requires 50 for that print, but if it was a less viscous material, it would require even less. So this is a significant reduction in required pressure, which is very good for cell viability and proliferation, because you wouldn't want to kill all the cells while you're printing them, because then your scaffold would be useless. And one of the ways we reduce this pressure is with the shear thinning properties that Slesha mentioned. So these are some of the scaffolds that were printed using the system. You can see in the middle, it is cross-linked, and it's kind of hard to tell, but you can see how different layers are different colors of, I think the bottom is green and the top is blue. And then above that, there's a not cross-linked and a cross-linked version. So these are just different scaffolds showing that it can print different layers with different materials and transition between the two without stopping the print. So the materials used were alginate and carboxymethylcellulose. The CMC material is a thickening agent used to increase viscosity. And the alginate is really good because it's natural. It comes from seaweed, and it comes as a sodium alginate salt, which can be mixed with water to create the hydrogel. And since it's natural, it's very good for the cells. And it also is very rapidly cross-linkable with calcium chloride or other double valence salts like that. So that's really nice. You can see the cross-linking in the previous one, which basically adds structure and immediately solidifies the print so it can't just disperse all over like the top and bottom uncross-linked scaffolds. All right, so the 5 different compositions that were used were tested for the rheological properties, and one of the big ones is viscosity. The one that showed the least viscosity was 8% alginate and 0% CMC, because it didn't have that CMC to add viscosity. But surprisingly, this is not the best transition material. It's actually 6% alginate and 2% CMC, which is the second lowest viscosity. And all of these show shear thinning properties because of that downward trend in viscosity versus shear rate. So these are some of the spirals printed. And the printer that was used was a CellLink BioX bioprinter that was new over the summer. So that was really nice. And okay, so the 6-2 combination of alginate and CMC is shown here as having the lowest initial full and actual transition distance, which again is that difference. So this is very good, and the reason that it was that one and not 8-0 that had the best transition is most likely because 8-0 wasn't viscous enough and actually mixed more within the transition chamber, which took longer for it to switch from one to another because it mixed so much, whereas the slightly more viscous 6-2 was able to switch from one material to the other without mixing as much. And then once it got more and more viscous towards the completely CMC material, it was too viscous, so it required more pressure to print at the same volume by speed, and that just slowed down the transition because there was less material leaving the nozzle at the same speed, so it increased that distance. In the future, I've been working on the transition distance for different nozzle diameters and for high viscous compositions. I've also been working on different material compositions for fabricating large scaffolds, which some use pre-crosslinking and some use post-crosslinking, but that's being looked at because, as you can see, a lot of those scaffolds are very small. I think most of them were 30 millimeters, and they don't get very tall because there's not a lot of structure, because if they do get too viscous, they require too much pressure to print. We're also going to analyze the pressure distribution along the nozzle length with finite element analysis, determine shape fidelity of scaffolds, and hopefully start fabricating scaffolds with encapsulated live cells with multiple cell types. I'm also going to be designing a bioreactor, which will mimic the environment of the human body. So once the scaffolds with live cells are printed, they can be placed in this bioreactor, which keeps a constant flow of nutrients or a varying flow and eliminates or takes out the waste from the scaffold so that the cells can grow and can be observed swelling and growing. And that can be tested to see how well the cells react to different materials. I would also like to start experimenting with different gelation methods of alginate, such as freezing or other cross-linking methods. And something that is a little different, but is also of interest to me, is researching the porosity and custom drug delivery techniques. So as I discussed earlier, the scaffolds can be printed with different porosities for different patients. This can also be used for drug delivery. So if someone needed a drug to be absorbed or metabolized slower or faster, or at varying speeds, the porosity of the pill could be altered in order to change this and customize drug intake for different patients. These are just some of the presentations and publications so far that we've participated in. And then I'd like to thank Keene State College, the Speedy Program, New Hampshire Biomade, and New Hampshire Embry. And then does anybody have any questions for Slesha, I, or Dr. Habib? Thank you.
Well, let me jump in there. Yes, and I see people clapping and I just wanted to say before the questions begin, really, really awesome presentations. Terrific job. A consistent thing we noticed through the semester is that at the end of presentations on Zoom, it's quiet because it's Zoom, which is a shame because when a presentation is really impressive, it deserves an audible applause. Nice job. And another sort of compliment that I saw in the chat was both of you had sort of background on 3D printing and your topic in a way that actually made people understand from zero to understanding in just a couple of minutes. That's a real trick, bringing people right along without a ton of prior knowledge. So really nice job. Having said that, I guess I'd want to hand it over to questions. And Professor Habib, do you want to start with those, or do you want to go to the board to see who's got questions? I see one. Jim Crayley has a question.
Yeah, thanks, Mike. First, really want to echo Mike's sentiments there. Fantastic presentations, really clear. Just amazing to see all the data, the progress that you've made. And so congratulations for that. Carter, what a great way to finish with a long future work slide, right? All these experiments that you have lined up. It's exciting to think about you making progress on that and other students joining the efforts there too. I guess I'll throw two questions at you, and whoever wants to answer them is great. The first is about the substrates that you're printing the different structures and scaffolds on. Are those always the same? Is that material, or even the what's physically there, whether it's flat or there's wells? Is that something you have or will experiment with? Do you think about the chemistry that's there? Is it silated, or is it glass, or plastic? Are you keeping that variable constant for the moment? And then the other thought I had is, have you wondered about how some of these structures can retain fluids? I know working with cells is the next step, but the ability to retain a volume, maybe retain a concentration of something that's there, whether you can measure that, and whether you could measure it leaking into the material itself. So thanks again for a great presentation.
I think I can answer the first question that you had about the substrate. So right now we are trying to, as much as we can, we are trying to make everything consistent with, even with the concentrations and all the materials that we've been using, along with the equipment and stuff. I think we've been using glass petri dishes, and but in past I personally have used plastic petri dishes, and the results are using both, like they just have like very separate, like you can see the difference of using it or not. But the scaffolds tend to stick more on the plastic, I would say, rather than the glass petri dishes. But yeah, I think we would be open to using more substrate in the future if we have the chance. But right now we are just trying to make it more consistent with whatever we do.
Yeah, I use glass for all of my prints, but I have tried different methods of cross-linking. So I've tried printing into a calcium chloride bath, and also allowing that to dry. So you're printing on a dry calcium chloride bed, kind of. And I'd like to look more into that. As far as the absorption or the ability to hold liquids, I think we looked into absorption rates a little bit, but I mentioned that I wanted to try different gelation methods for alginate. So I was interested in kind of studying micro or macro porosity of the scaffolds and how that would affect cell proliferation, as well as how it can absorb nutrients and affect the area of the scaffold. So yeah.
Great answers. Thank you both.
Yeah, I just wanted to add a little, right? So yeah, so I did some swelling test, and it is honestly in our bucket list, or in in our future work, right? So we didn't spend that time on swelling because we didn't start the cell encapsulations, but we have to start it as soon as possible before just starting the cell encapsulation.
So I have a question, a rheological question, and I also have another question about the prototypes, and that is a shear thickening liquid. I'd never heard the distinction. I'd actually never heard, but it's obvious to me, you know, that cornstarch is a shear thickening liquid. But also it seems to me that shear thickening liquids must be pretty oddball. There must not be a lot of them, because it's cornstarch is the only one I can think of. Are there lots of occurring shear thickening liquids out there in the world?
Um, I I have done I have not done that much of research into it, but when I went, because this was like a very foreign term for me too, but I went like online and saw like shear thickening materials and stuff. But I think there are quite few materials like cornstarch, and I think certain kind of
Mm-hmm.
slimy materials that you would just hit and it just feels like a concrete. Like it is as same as whenever you know, like I don't know, if a person is like jumping from like a very high altitude in the water does not feel like water anymore. It's just
Right.
like a concrete, so you can actually like kind of compare it to that thing. And yeah.
So that's a really interesting point. So at some point, shear thickening happens with a variety of liquids, including water.
Yeah.
Okay.
Yeah, again, it also depends upon like what kind of force you are putting into it,
Sure.
like the intensity of the force.
Sure. Like,
I don't know a lot about this, but I think there's also smart liquids that kind of show these different properties under stimuli, like electrical fields and stuff, that can actually change their viscosity. And I think they were used in a couple different car suspensions that were released on the market. So that's kind of cool.
yeah, no kidding. So my other question is about the prototypes, Carter, that you were talking about. And you went from a prototype that you printed yourself, you know, with nozzles and things like that, to getting some medical equipment and sort of putting them together in an interesting way with valves and et cetera. And I thought that was a cool idea. And then the other thing that I thought seemed like a cool idea is you wanted to measure distance in the transition, and instead of making it a line and you know, going across the line and seeing how long it was, you made a spiral. And at first I was thinking, why did I make a spiral? But it strikes me that you probably made a spiral to allow the printer to work within its radius. So my question is, how did you come up with this? These really good solutions to some really tricky problems. Yeah.
Thank you. So I did a lot of different prototypes, and kind of solved one problem at a time and just kept finding problems. And a lot of them had to do with just the technology available. So as I said, the printer we had was pretty old and couldn't really get very precise. We actually just got a lot of new 3D printers, and we just got a resin laser-based 3D printer, which is much more accurate. And so I probably could start doing more prototypes that were more higher resolution, but at the time it just wasn't really possible. So the best option was to buy pre-made medical components that were already precise.
Mm-hmm.
And an interesting part about that was that to print that whole system, I actually kind of had to trick the printer into thinking it was doing something else, because it's having pressure come from the two side print heads. So it thinks it's printing out of the side print heads. So when it prints, it actually lowers the two sides. But the nozzle system's connected to the two sides and they're both calibrated to the middle. So it thinks it's printing from one side, but it's actually the middle that the pressure is going to. And then the spiral I just thought would be a lot simpler because I didn't know how long the transition was going to be at the time and didn't know if I'd run out of material. And it's also a circular petri dish. So I was thinking, why would I make it a square or like a bunch of squares that had different measurements because then I'd have to add all those different measurements together and measure the length of each thing and where it stopped. And I was like, I know the speed that the printer's going, and if it's a spiral, it's constant. So if I just multiply speed by time, I can find the distance really easily. So I just plug it all into an Excel sheet and it does that. Yeah, so I kind of just went with what was easiest for me.
I promise you that wouldn't have been easy for me. That was really good explanation. Sort of a related question in the chat. Wes Martin is asking, what are the limits of scale or size in printing biomaterials? Could you actually print a whole liver right now, or are 3D printers limited in how big they can make an object?
Over the summer, we printed a full-size ear, and that was a little tricky just because the weight of the top materials keeps pressing down on the lower ones. It is possible to print fairly large structures because you can increase the viscosity as much as you want till it's basically solid. So you could have a solid that has shear thinning properties so that it can print. The problem with that, and the reason we aren't researching that, is just because it would kill all the cells to use that much pressure.
Sure.
Yeah.
So
Just want to add a little here, like, so that's why we are basically trying to optimize like the concentration or the viscosity of the material, at the same time the viability of the cell, so we are basically trying to confirm the printability or the shape fidelity of the structure we are designing, and at the same time we are just allowing some safe house for the cell so that it is not getting that much pressure and it is becoming tight.
yeah, it seems like a real big puzzle with lots of variables. And that's another one of the very interesting points that's come across in all of your talks. There's another question in the chat. It says, in the future, would the long-term biodesign goal be to make each organ custom made for each patient? Oh, that's a really cool question. Yeah.
So basically, the main reason for doing 3D bioprinting that involves this issue. So even when we're like organ transplantations and stuff, when people like transplant organs and like get like organ donated, and the body cannot, like, there are like a lot of cases where the body does not just accept the organ and stuff. So yes, this is something that 3D bioprinting has been like trying to solve, because the way it works is while designing the organs, we take an MRI or CT scan of the organs that the patient needs or has already, and the entire designing of the organ is like, it revolves around being very host specific. So early on in the design process, we make sure that it is very specific to the host. And even then, because it's still an experiment and we still don't know if it's going to work or not, if the host body is going to accept it or not. But that is, yes, the answer to the question is a big yes, because that is the kind of challenge that 3D bioprinting is trying to solve.
Sure. What do you think the first bioprinted organ or body part will be? Bone? And people always talk about liver. Is there an easy target, an easy next one? Tooth?
So Carter, do you want to jump, or? Because I'm just teaching them in the biofabrication course this one. I think they know the answer, at least I know that.
Yeah, so one of the big ones that people are starting out with is bone, because it doesn't require vascularization as much. And that's really tricky to do. And people are trying to do that in different ways, but to print like these really micro blood vessels within an organ is really tricky. So You can use different cells, and people are trying to do this in different ways, but bones don't need as much vascularization. So that's what they're starting with.
Other questions? Got comments. A lot of them are, wow, you guys are smart. Kind of comments. I think that's apparent and definitely affirmed by people in the audience. Does anybody, Wes, you have a comment on
I
the?
think I see a question in the chat, and I would love to answer that.
Yes.
So it says, is shear thickening and thinning heavily dependent on temperature?
That's Wes's question. Yep.
And it's very interesting for me because like this is one of the mistakes that I did while I was doing the research. Like early on, I did not know that we were keeping the materials like a consistent amount of material, the concentration, composition, and all the equipments that we were using consistent. So the one of the problems that we had at the beginning of making the biomaterial was that for very viscous material, it was so hard to stir, and we did at that point we did not have a stronger stirrer. So I just approached Dr. Habib with this problem, and he said, "Oh, you know something? Just use because the stirrer had adjustable temperature switching, like it could like heat and lower down the temperature and stuff." And he said, "Why don't you use that?" And I started using that, the temperature thing, and it actually worked better because it lowered the viscosity, and it was so easier to like stir it overnight. So the answer to that question is yes. The shear thickening and thinning behavior of a biomaterial is highly affected by the temperature. And the reason behind it is because the way I see temperature is like a disturbance. So whenever there is a biomaterial, or whenever there is anything that is closely knit, that it has a chemical composition or like a molecular formula, when you introduce heat to it, there are certain bonds that just like disrupt. So whenever, it's like staying very compact and it's a solid, but whenever you introduce heat, temperature, there's like a certain disturbance. And when the molecules fall apart, it shows more of a liquidous characteristic. So, which is a change in the viscosity. So, yes.
Great answer. Yes, that was the question I was aiming at. So fantastic. Does anybody have other questions? Professor Habib, would you like to make any comments or add any questions to your students?
Okay, I just want to say it was a great presentation. Yes, and yeah, honestly, every time, because Slesha presented in AEC last Saturday, and Cartwright is already presented in Biomed meeting and in BRI meeting, so they were doing great, and today they did the same. So I'm very happy that I got to grade a student as my research student. And honestly, we have like a big plan for this summer, and we are definitely looking forward that thing.
Yeah, well, I definitely see great things for you guys, and it should be a great summer. And with that, I think we can call it another evening.